afternoon, ladies and gentlemen, and welcome to this H3 Symposium, where we hope to go back to basics about understanding intravenous fluid therapy. This is quite a novel symposium in that it is a mixed audience of scientists, physiologists, clinical researchers and clinicians, and this symposium has been jointly funded by the Physiological Society and the Association of Anaesthetists of Great Britain and Ireland. And I'd like to acknowledge their most generous support in helping us put this meeting together. I'd also like to thank Sarah Bundop from the Physiological Society for all she's done in managing us all and getting us here for today. Why are we here and what are we doing? Well, I can explain that by showing you this picture of these two very eminent physiologists, William Bayliss and Ernest Starling, and relating them back to the first medical research committee that was established during the Great War. And it was established to investigate treatments for wound shock and allied conditions. William Bayliss was Professor of General Physiology at University College London at the time and had been doing work on the use of gum arabic solutions in intravenous injection and sustenance of circulating volume. And he presented a paper to the Physiological Society on the 25th of March 1916, and this was published, and it's probable that he was co-opted onto this committee because of that work. Ernest Starling, as I'm sure you all know, was working profusely in publishing in terms of the work of the heart, in terms of the law of the heart, and also the exchange of material across the capillary wall and what we've come now to know as Starling's equation. And both of them served on the committee at the same time, and both of them chaired that committee in succession. They also worked together at UC, and they were brothers-in-law. Bayliss, when he was co-opted onto the committee, decided that he would go to the battlefields of the Somme to gain some experience of what was happening in the field. It took him a while to get there because there's an anecdote that on his way there he managed to slip his minder and he ended up getting arrested in France as a spy. Well, eventually he did get there, and what he saw and what he learned and what he helped with were the experiments that were conducted in the field at the time of the Somme battles. At that time, sodium chloride, 9% saline, was routinely used in resuscitation, and as you can see from the graph that Bayliss later published, it didn't really do much good. Whereas on the right, the introduction of gum acacia or gum arabic to the saline solution really did seem to manage to maintain the circulating volume and so systolic blood pressure. This process was adapted by some practitioners at the time in the battlefields, but it wasn't uniform, partly because I think people didn't understand the rationale for the addition of the gum acacia, and also the gum acacia that was supplied was of variable quality, and so results were themselves variable. Bayliss did write up this work and published it at the end of the war, but I think he never got the recognition that he deserves for what was really some very innovative science done over a very short period. Well, 100 years on, we're here again, and I think again I don't have to remind this audience that there is some controversy around the use of infused colloids and crystalloids, and that some of the use of semi-synthetic colloids such as hydroxyethyl starch have been challenged. And many of you will probably have seen Christiane Hartog's article that was published last week in the BMJ, and she hit the cover with it too, with a very nice review of some of this controversy. Also, I think that the physiological mechanisms by which these additives have their advantageous and sadly disadvantageous effects remain uncertain and unresolved. And finally, I think that part of this may be a consequence that the topic is inadequately covered both in undergraduate textbooks and in our preclinical and clinical teaching. And so Tom Woodcock and I, who are co-organising this meeting, decided it was time to bring some of the experts in the field together and to bash it out. And that's exactly what we're doing today. Charles Michel is Emeritus Professor of Physiology at Imperial College, London, and sometime Fellow of the Queen's College, Oxford. He's published, I think, well over 100 papers on microvascular fluid exchange and the nature of the interface at the capillary wall. His contribution to the field has been recognised by numerous awards, and the Malpighi Medal from the European Society of Microcirculation, the Annual Prize Lecture from the Physiological Society, amongst them. He's an honorary member of the Physiological Society, the American Physiological Society, and the British Microcirculation Society, and we're very proud of that. I've known Charles for an awful long time, and I've had the privilege of working with him both in Oxford and in London, and I think I can honestly say that I've learned from him most of what I know about the capillary wall and exchange across it. But I think I'm going to learn more today. So Charles, please.
I hope you can still hear me, because I just dropped the... oh yes, it is actually. Geraldine, first of all, thank you very much for the very kind introduction. I think most of what you learnt me was doing things yourself in the lab. Discovering what I suggested might happen was in fact not the case. Now, let me find this. Okay, right, and this is the point. Right. I should begin by, first of all, apologising that what was, I think, put out as my title, I've reduced somewhat. Originally, I was going to talk about Starling's Principle, classical and revised, and also plasma volume regulation. But in fact, I realised that with only 40 minutes to talk in, I would do well just to go through the first two parts of this. And then in the last 3 slides, I'll just show some modelling which may be relevant to the application of the so-called revised Starling Principle to fluid therapy. Put out as my title, I've reduced somewhat. Originally, I was going to talk about Starling's Principle, classical and revised, and also plasma volume regulation. But in fact, I realised that with only 40 minutes to talk in, I would do well just to go through the first 2 parts of this. And then in the last 3 slides, I'll just show some modelling which may be relevant to the application of the so-called revised Starling Principle to fluid therapy. Now, my first slide is actually a picture of Starling in 1896, the year in which his paper on fluid balance, his famous paper, his classical paper on fluid exchange, was published. And it was really the culmination of 6 years' work, or 6 years' interest, and most of it involved in direct experimentation on the subject. In 1890, the subject of lymph formation, as it was called, was quite controversial. In the 1850s, Ludwig and Noll had suggested that lymph, interstitial fluid that is, and lymph, had been formed by, was formed by ultrafiltration in the capillaries. Now, many people disputed this, largely because if you raise the arterial pressure, you don't necessarily see an increase in lymph flow. And this was not understood why this should be if, in fact, Ludwig's theory was correct. In fact, some people did understand, and some of these people were really quite influential, including the professor of physiology in Breslau, now Wrocław in Poland, Julius Koenheim. And he has a perfectly modern explanation of what was going on, namely the fact that the pre-capillary resistance would in fact raise the arterial pressure if you had generalized vasoconstriction, and actually lower the capillary pressures downstream. But many people didn't understand this. And in fact, after Kernheimer died, his former colleague Heidenheim, the professor of physiology, I regret to say, in Breslau, he proposed that lymph was actually secreted by the vascular endothelium. And he carried out a series of experiments which he believed proved this. In fact, they didn't prove it, but what they seemed to show was that filtration didn't work. Now, this paper was published in 1891, and in 1892, Starling and his wife went over to Breslau for 3 months in the spring and summer of that year, and they worked in Heidenheim's lab. And Starling did some work on the distribution of peptides, peptones, I should say, between the blood and the lymph, which also suggested there might be secretion. But when he came back to London, and he was lecturer in physiology at Guy's at that time, he began repeating Heidenheim's experiments. And he began doubting the significance of them with respect to the filtration hypothesis. And he made occasional trips north of the river to University College, where he set up a friendship with William Bayliss, whom Geraldine has referred to, and was later to become his brother-in-law. And they began collaborating in a way. And during the period of 1892 to 1894, they carried out some really beautiful, old-fashioned, beautiful mammalian physiology experiments in which they showed that far from demonstrating that secretion of lymph occurred at the capillary bed, all Heidenheim's experiments could be beautifully interpreted on a filtration hypothesis. Well, Starling also started thinking of movements of fluid going into the blood, directly into the blood. At that stage, most people had believed that fluid, once it left the plasma, either by filtration or secretion, could only get back into the blood via the lymphatics. But Starling thought there was evidence against this. And in fact, Kernheim, who I'd referred to previously, in his lectures in General Pathology, he actually suggests that this happens after hemorrhage, when in fact the blood is diluted quite rapidly. That is, that you get an anemia, and also you get a dilution of the plasma proteins. So Starling then noted this. He also noticed that fluids could be absorbed directly from the peritoneal cavity. And he then carried out a series of remarkable experiments himself, in which he perfused the isolated hind limb of a dog by a mechanical business of merely collecting the blood as it came out of the vein into a beaker, and then pouring it into a funnel which was going into the artery. And he recirculated the limb in this sort of way. He had no pumps or anything to do this. It's a very crude system. And he found that if he injected saline into the muscles of the dog, this saline was absorbed into the plasma, and he could actually measure the fall in protein concentration which resulted. However, if he injected serum into the muscles of the dog's leg, then in fact it remained there. There was no absorption at all. And this led him to think that perhaps the proteins in the plasma were exerting an osmotic pressure which was opposing uptake into the blood. And he measured he was the first person to actually measure the osmotic pressure of macromolecules. And he did this 1896, and he got a value for the osmotic pressure of dog plasma which agreed reasonably well with the estimates that he and Bayliss had made for the capillary pressure. And so he was led to his hypothesis, which should appear now when I click. Oh, there we are. The osmotic attraction of the serum for the extravascular fluid will be proportional to the force expended in the production of this latter, so that at any given time there must be a balance between the hydrostatic pressure in the blood, in the capillaries, and the osmotic attraction of the blood in the surrounding fluids. Now that is Starling's hypothesis in his own words, and that is as most people see that he understood it. In fact, as we shall see a little later, he understood a bit more. Now, Starling was an immensely prolific writer. He wrote reviews, gave lectures, and which were published, of course, and he also wrote textbooks. And he propagated his ideas in this way, but he did no more experiments actually on blood-tissue fluid exchange. He did actually improve his measurements of osmotic pressure, and he did see the significance of them for renal ultrafiltration. Again, something that was controversial at the time. But even as late as 1911, Luciani's textbook denounces, well, I say denounces, it doesn't really denounce, but it actually says that Starling's hypothesis is most unreasonable, it's much too simplistic, it's mechanical, it goes back to the mechanists of the 19th century. We don't want this sort of rubbish. And I've heard that sort of thing said more recently at meetings of, not of the Physiological Society, but at international meetings about what simple ideas of mechanisms which are being proposed. Most people I think were becoming convinced, but many people remained unconvinced, really until 30 years after Starling's original paper. And the evidence that really swayed everyone was the work of a University of Pennsylvania medical student, Eugene Markley Landis. And he did this. He did two remarkable things. In the evenings and weekends, during the time that he was a medical student, he first of all devised a method for cannulating the capillaries in the frog mesentery and measuring the pressure inside them. And secondly, he developed a most ingenious technique for measuring the filtration of fluid through the walls of the capillaries per unit surface area. And he couldn't vary the pressure in the capillaries, but he found that there was a distribution of pressures which is sufficiently great, and when he plotted this against the filtration rate, he got this very nice linear there we are, linear relationship. Filtration rate, filtration up here, reabsorption down here, capillary pressure, this nice correlation between the two. And furthermore, this zone of pressure here correlated with the colloid osmotic pressure, which H.G. White had measured in frogs of the same species, H.G. White being interested in the kidneys of the working of the kidney and glomerular filtration, and work was being done just at the same time, also at the University of Pennsylvania, by Chambers, of course, and Richards. And it was fortunate for Landis that this work was done, because it all seemed to fit together rather nicely. Landis also described this relationship by a simple equation, namely that filtration and reabsorption is proportional to the difference between the capillary pressure and a constant, and this constant had a value very close to the colloid osmotic pressure of the plasma. Now, I would like to emphasize Landis's experience, because he was the first person to introduce this relationship, filtration rate against pressure, and the importance of the colloid osmotic pressure of the plasma. Because I think this lays the foundation for the classical interpretation of Starling's hypothesis, which became Starling's principle, and is the diagram which I'm going to use throughout most of the rest of the talk in different formats. Let's consider really now rather quickly how Starling's hypothesis became Starling's principle. Well, here we've got the statement in words, and then we've got Landis's statement in symbols. PC, the capillary pressure, PI, the interstitial fluid pressure, pi C, the colloid osmotic pressure within the capillary in the plasma, and pi I, the interstitial fluid colloid osmotic pressure. And Landis's measurements seemed to show that the interstitial fluid pressure and the interstitial fluid osmotic pressure were not really significant. But in fact, Landis himself, if you read his 1934 review, you'll see he was uneasy about that, and he thought that there were certain circumstances when these would become important, and he wasn't wrong here. Now, the next set of advances occurred really in the 1940s. And that was when Pappenheimer and Soto Riviera showed as convincingly as Landis had showed in the frog that you had the Starling hypothesis or the Starling principle operating in the perfused hind limb of mammals. But then a conceptual development occurred in 1951 when Staverman introduced looked at the theory of osmotic pressure across leaky membranes and showed that in fact you didn't get the full osmotic pressure, you got a fraction of the osmotic pressure, and that fraction was dependent upon the leakiness of the membrane. It was it was called the reflection coefficient, and it represented the fraction of solute that was reflected at the membrane during ultrafiltration through the membrane. And this idea was brought into membrane physiology by Kadam and Kochalski. There should be more things coming up. Here we are. Then in the 1960s and 70s, Guyton measured interstitial fluid pressure, found it was below atmospheric, much to everyone's surprise, and that it did change with filtration rate, though over a relatively small range in most tissues, the lung being a possible exception. And then people also measured the interstitial fluid osmotic pressure, and this was very different from zero, became very significant indeed. So that finally, if you put Starling's hypothesis in its most general format, which I've done here in the bottom expression, the filtration rate per unit area is proportional to the difference between the hydrostatic pressure gradient across the capillary barrier and the sum of all the osmotic pressures across the capillary barrier. We've got basically a statement of the same law of thermodynamics, and you can be fairly confident that this is Starling's principle rather than Starling's hypothesis. And just to show you how you can confirm Starling's principle, this is an experiment from my own lab. We learned how to vary the pressure in a single capillary over a wide range, and at the same time measure the filtration rate and reabsorption rate. By this stage, it's in the late 1970s, around about 1980. And we've got here a plot of the filtration rate, the absorption rate, against the capillary pressure in a vessel which was first of all perfused with a Ringer solution containing 8% serum albumin, and then another Ringer solution containing 2.5% albumin. And you'll see you get the linear relationship now in a single capillary, which Landis reported in a whole population. And you'll also see that the effect of the change in perfusate concentration led to a lateral shift, and the size of that shift was equal to the difference in effective osmotic pressure which the serum albumin exerted across the capillary walls. And that, I think, is the sort of classical picture. But it doesn't fit all the observations. It certainly doesn't fit many clinical observations. But one experimental observation which became clear to us about this time was that you could get a similar picture without changing the perfusate concentration, but merely changing the pressure at which a vessel was perfused. Here we've got filtration, reabsorption against capillary pressure, just as in the last slide. Now in this case, to determine this relationship, we perfused the vessel at a high pressure, at 35 centimeters of water, relatively high pressure. And we got a classical picture with the effective osmotic pressure very close to the osmotic pressure of the perfusate, which is here. Now if we perfuse the vessel at a low pressure, a lower pressure, between 15 and 20 centimeters of water, and we do this for 10 minutes or so, then if we redetermine this line, we get a shift from this position to this position. And now we get this line. So we've got the lateral shift without any change in perfusate. And of course, if Starling's principle is correct, which I believe it must be, the general statement, then we're changing something else. Either we're changing the hydrostatic pressure gradients or we're changing the osmotic pressure gradients. And we might now just go back and look at something that Starling said in 1896. He said, with increased capillary pressures, there must be increased transudation until equilibrium is established at some higher point when there is more dilute fluid in the tissue spaces and therefore a greater absorbing force to balance the increased capillary pressure. And with diminished capillary pressure, there will be an osmotic absorption of saline from the extravascular fluid until this becomes richer in proteids, and the difference between its proteid osmotic pressure and that of the intravascular plasma is equal to the diminished capillary pressure. He finishes up by saying, here then we have the balance of forces necessary to explain the accurate and speedy regulation of the circulating fluid. And what I believe he's doing here is he's moving from the acute changes in pressure and the acute shifts of fluid to the long-term regulation of plasma volume. In fact, he goes on. He's actually saying that here is a speedy regulation of the circulating fluid, the total plasma volume. Now, what he's saying in the first two statements was actually, some of it was known in the 19th century, but had been really not understood in this context. For example, Kernheim, again, I go back to, knew that if you raise the venous pressure, then the flow of lymph increased because he believed the capillary pressure was increased, and also the concentration of proteins in the lymph decreased. Now, by the late 1970s, when I started thinking about this problem, there were lots of equations which I could use, work I could draw on, in which I could calculate a curve of this kind, which is the concentration of proteins in the ultrafiltrate compared with the concentration in the plasma. And, oh, dear me, there we are. Here's the plasma concentration, And here we've got the fall in concentration as we increase the filtration rate. Now, this of course would only be the concentration affecting fluid balance in a steady state situation, because once the filtrator got downstream it would mix by diffusion with the interstitial fluid around it. So I thought. But what it implied was that the concentration difference for proteins across the capillary wall would also vary with filtration rate. And again, if the difference was the plasma concentration was 25 millimeters of mercury, then in fact if we had no filtration, ultimately in the steady state we would have diffusion equilibrium across the capillary wall and you'd have no difference in concentration. Then as you began to filter you'll get a more dilute concentration in terms of protein in the ultrafiltrate, and you could go on, and then if you raise the pressure even further you get a higher filtration rate and you begin to approach the limiting value, which in fact would be the difference in concentration multiplied by the reflection coefficient. So this implied that there should be in the steady state a non-linear relationship between filtration rate and pressure. And Mary Phillips and I set out to prove this, and we did an experiment which has been adapted by Rod Levitt for his textbook, and I got this diagram from his textbook. Well, no, actually I think we used it in a review we wrote, where again filtration, absorption, capillary pressure. We started off perfusing at high pressure, obtained the classical linear relationship. We then held the pressure down here and watched what happened to the filtration rate, and we saw the reabsorption rate, and we saw that with time it diminished and eventually finished up at effectively zero. And then if we raise the pressure, we would go up here, a shift in the effective osmotic pressure across the capillary wall in this fashion here. And so we'd been able to reconstruct the observation which had been made earlier. So this had the interesting implication that there could be no reabsorption in the steady state. And that is, I believe, true in all the tissues where the interstitial fluid is formed as a microvascular ultrafiltrate. However, there are other tissues of the body where we do have steady state reabsorption, for example in the kidneys and also in the gastrointestinal tract. But here there was a let out, because here the interstitial fluid was formed largely by a protein-free secretion of the neighbouring epithelial cells. And this kept the protein concentration of the interstitial fluid down, and we could have continuous reabsorption. Another example occurs in the lymph nodes, because here we have a constant flow of prenodal lymph over the capillaries, the vascular capillaries in the lymph node, and it is possible to have steady state reabsorption there. Now, a question which rose in the 1980s and early 1990s was, how well do the Starling pressures balance? Because the plasma volume remains more or less constant, so we must have rough balance between the Starling forces, if this is the only mechanism of importance in determining the plasma volume. Well, one way to do this was to take from the literature values which would fit into a simple manipulation of the Starling equation. Filtration, absorption. Next, if you had balance, you would have no filtration. When that happens, your pressures, your hydrostatic pressure and effective osmotic pressure would balance. So you can have a statement where when the capillary pressure, when filtration was 0, capillary pressure there would be equal to the sum of the interstitial fluid pressure plus the effective osmotic pressure across the capillary walls. And there were lots of measurements in the literature by the mid-1980s which allowed one to make estimates of PC0 by calculation. There were 1 or 2 direct estimates of capillary pressure, and so it was possible to make comparisons. But in the mid-1980s I tried this, and they didn't balance, but I thought that, well, perhaps one measured pressure in capillaries in which was a brisk flow, because you could see them better, and you were probably not, your sampling was not representative. But my very good friend Rod Levick was much bolder, and he estimated, compared PC0 for the venous pressure in various tissues, or the venular pressure. And here are the results he obtained. Actually, most of them are the ones he put together, with 1 or 2 I added when we wrote a joint review together. So that's why it's from here. But here we've got the venular pressure. Here we've got the calculated sum of the Starling pressures opposing filtration. Most of the points lie on the upper side of the line, indicating net filtration. These points fit in with our hypothesis about steady-state reabsorption. Then they're on the other side of the line. And even more striking is that in intestinal mucosa, during fluid uptake, the point lies here. When there's no fluid uptake, the point moves over to here. So that fits in very nicely with the idea, but it leaves you with the problem of there's not a balance at all. You've got an excess of pressure, even if you use the venous pressure rather than the capillary pressure. And this, much to Rod Levick's delight, torpedoes what is the classic diagram for the paradigm for teaching Starling's hypothesis, the famous picture of capillary pressure declining along the length of the capillary, with a higher than the osmotic pressure of the plasma proteins in the arterial end, and reabsorption in the venous end. In fact, we know that these pressures all lie well above here, or at least they would appear to lie above here, and so this paradigm really has no basis in fact, and we can forget about it. We should actually drill it out of the textbooks. That's what Rod Levick thinks. He feels very strongly about this. But there is a problem, actually, because if we do look at these differences in between the calculated balance pressure and what has been measured, you do have a net pressure driving fluid into the tissues. And the question is this the necessary for filtration? Well, the answer is certainly not, because 3 millimeters of mercury excess pressure in muscle, and in some muscles the pressure difference is greater. That will give you a lymph flow of 10 litres per minute. In fact, estimates of lymph flow in humans between 2 and 4 litres per minute a day. So, 10 litres of lymph per day, it would predict, not per minute. So 2 to 4 litres per minute is what you actually measure. Well, there is a discrepancy, because fluid is actually taken up in the lymph nodes, and quite a significant amount is removed from the prenodal lymph in this way, it's believed. But nevertheless, you can't account for things in this way. And that led to a heated discussion we had at a meeting on Starling's hypothesis in 1996. And the question is, what is the explanation? And to really get to the basis of this, one has to say, is one doing are we doing the sums correctly? And both Shelley Weinbein and I thought we were not. And the clue is the way in which fluid and proteins cross capillary walls. And for 60 years, more than 60 years, we've known that there are at least two pathways. One for water and small hydrophilic solutes, which is not available to larger macromolecules. And this is illustrated in this more recent paper in which the clearance of proteins and between the plasma and the lymph has been plotted against the molecular radius of the protein concerned. And you see it has two components. There's a sharply falling component here and a more slowly component here. And the sharply falling component is described as being the small pore pathway, and the more slowly following is the large pore pathway. Now, the idea of pores, as we'll see, is controversial, but it has some merit to it, and it enables one to do calculations. But the idea that you have two separate pathways immediately makes one query the simple calculation that Levick had done. And if you separate out what are the mean osmotic pressure, that is, which arises from the mixing of the two flows from the large pore and the small pore pathway, and what is actually coming separately from the small pore pathway, you find that the effective osmotic pressure is much greater across the small pore pathway. I say much greater, it's only 3 to 5 centimeters greater, but that's quite significant in these terms as we saw from the original paper. And it means that we have a significantly high effective osmotic pressure, and that appears to bring things much closer to a balance. Now the question is, how do you get this separation occurring? Well, the small pore pathway is really now thought to be the interstices of the glycocalyx on the luminal surface of the endothelium. And here's a picture of the glycocalyx from a frog mesenteric capillary. It's a deep etch, rapid freeze deep etch picture, and Dr. Arkle will be telling us much more about the structure of the glycocalyx later on. But if that's the filter, what happens next to the fluid? Well, the fluid then has to go through the intercellular clefts. So it's actually channeled to a much narrower space. The large molecules either are carried by transcytosis or through occasional transcellular channels, or possibly even occasional widened intercellular clefts. But why can't the protein diffuse back up here below the glycocalyx? And Shelley Weinbein and I had the same idea that in fact not only is the fluid channeled into the clefts, but it has to be channeled through the breaks in the tight junction. And this means that if you have even a tiny flow, the velocity when it gets channeled through narrower and narrower spaces increases, and so you get a flow which is high enough to prevent back diffusion. And the back of the envelope calculation which I did suggested this quite definitely, and a detailed mathematical model by Weinbaum and Hugh confirmed this. And just to give you some idea of different capillaries, here's a frog mesentery, here's rat mesentery, here's rat heart, and here's skeletal muscle. So you've got very much a narrowing of the path through the intercellular cleft. Shortly after this was published, Roy Kerry said to me, "I know how to test your hypothesis," and he collaborated with Weinbaum's group, but I think the experimental work was largely done in his own lab. What they did was essentially perfuse a single capillary, measure the filtration rate and absorption rate in that capillary with the mesentery covered with protein-free solution. They then covered it with a solution which had the same protein concentration as the perfusate, and they waited, and had a fluorescent tag to this, and they waited until the concentration reached equilibrium around the vessel. And below here we've got the intensity of the signal from the interstitium, and you see that after 77 seconds it's down here, but it goes up 800 seconds, and round about getting on for 20 minutes you've got more or less a steady state arrangement, and the concentration appears to be right up to a micrometer around the vessel wall. And here's one of the experiments they did, again going to this the relationship between filtration rate and pressure. Here's the standard linear relationship with an intercept here when there's no protein in the external solution. We now add the same concentration of protein to the superfusate that's outside the capillary as inside the capillary, and you would expect on the classical Starling hypothesis to get a relationship here. In fact, the shift is relatively small to here. Now that, in a nutshell, is the revised, so-called revised. It's really just a development of Starling's ideas to the steady state, and also the location of where things are. I've got, sorry, three slides to show which I think might be relevant to fluid therapy. The first is, here's the steady state relationship, filtration rate against plasma, and where do the pulmonary capillaries sit? They sit down here. So this means that they're well on the flat part of the steady state relationship. And that means if you raise the pressure in the pulmonary microcirculation, you get a rapid increase, but that's followed by very rapid decrease, because as it turns out, the lung capillaries move from a transient to a steady state very quickly. Similarly, if we look now at the question of infusion of a crystalloid solution, you start at a normal capillary pressure here, then as you infuse the crystalloid solution, you'll dilute the plasma and you'll get an increasing filtration rate out of the vessels. If, however, the pressure is reduced down into this region, you'll have a very much smaller increase in filtration rate. And this I've modelled on a fairly simple sort of model, but it's to show you what will happen to the plasma volume. It's assuming that most of the capillaries in the body are like muscle capillaries. And above we see the changes in plasma volume which occur with time when the pressure is normal or raised, and here when the pressure is reduced. Now, this I think is interesting from the fluid therapy point of view, because if you try and look at the effects of infusing fluids, but you keep the capillary pressure constant, a normal sort of value, you'll get the wrong answers, because in a shocked patient you'll have intense vasoconstriction in the muscle, a much lower, much reduced capillary pressure, and the infusion of crystalloids would follow the lower panel and therefore be retained in the circulation for very much longer. My next slide is just to remind me that we don't just have muscles which are capillary beds which obey Starling's hypothesis, but there are capillary beds like the spleen and the liver where you have constant leakage of protein, and so they are going to have a major effect when you come to look at the regulation of plasma protein concentration and plasma volume. And finally, here's my summary, which is that first of all, there's nothing wrong with Starling's principle. We got it right in the classical form. Secondly, if you move to a steady state, then in some tissues where the interstitial fluid is formed entirely by capillary filtration, you can't get reabsorption. The reabsorption attenuates. It's a transient phenomenon. And finally, the relevant differences in hydrostatic and osmotic pressure are those across the endothelial ultrafilter, which is believed to be the glycocalyx. Thank you very much.